Because metabolism can be so, well, messy, it's common to break it up into quote unquote pathways. So we can talk about glycolysis, say, the breakdown of glucose, as if it were an isolated pathway. However, we have to remember that that's taking place in the context of this bigger picture. And glycolysis is connected to other processes, things like lipid metabolism. Even glycolysis, even that glucose from glycolysis, that could be coming from glycogen breakdown. It could be coming from the ingestion of glucose. We have amino acids come into play at various points in the chain. All of these, however, are kind of congregating at this end result, the electron transport chain. The electron transport chain is actually where you're going to get the energy payout from catabolism, from the breakdown of molecules. And spoiler alert, none of the carbons from the glucose, none of the carbons from those amino acids, none of the carbons from those lipids are actually going to go into the electron transport chain. None of them are actually going to be in the location where you're going to be making the bulk of your ATP. All that's going to be there is the electrons. And the electron transport chain does not care where those electrons came from. Kind of like how we can talk about ATP, and ATP can be spent by all these different enzymes, and those enzymes could care less where those ATP were generated from. Did the energy for them come from glucose? Did it come from proteins? Did it come from lipids? Who cares? You've got this final product you can use anywhere. Similarly, the electron transport chain, all it cares about is the electrons. It doesn't care where those electrons came from. And so by starting at the bottom up, starting with this electron transport chain, starting with how do we make this energy from these electrons, then we can better understand, okay, well, how exactly are those electrons being generated? It comes down to oxidative reactions, oxidative processes, oxidative pathways. So remember our redox? Reduction is gain of electrons and oxidation is loss of electrons. So you can remember oil rig. Oxidation is loss of electrons and reduction is gain of electrons. This is connected directly to thermodynamics. The movement of electrons from something with a lower reduction potential to something with a higher reduction potential is energetically favorable. When we say energetically favorable, think negative delta G, think release of energy. Now, be careful because when we talk about reduction potential, here we're going from a lower reduction potential to a higher reduction potential, and that's going to be favorable. Normally, when we talk about thermodynamic stuff, we're talking about going from a higher thing to a lower thing is favorable. But in this case, lower reduction potential means you don't really want those electrons, whereas higher electron potential means you really want those electrons. If something really wants electrons, it's going to be more thermodynamically happy if it has those electrons. And so giving it those electrons is going to make it happy. It's going to release energy. So passing electrons from something that wants it less to something that wants it more is going to be energetically favorable. You can then use the energy that's released to do cool things like making your ATP. And so this is why we see that the bulk of our catabolism, our breakdown of biomolecules for energy is going to be happening through oxidative processes. The more reduced something is, the more reduced your fuel is. So the more electrons it has. So remember, reduction is gain of electrons. So if we say something is reduced, we're saying it has a lot of electrons. If something has a lot of electrons, it has a lot of electrons to give. It has a high ability to be oxidized. So if we say like a fat, a lipid, that's going to have a lot of hydrocarbon. It's going to have a lot of saturation. It's going to have a lot of electrons it can pass off, as opposed to something like our glucose. Well, here we already have done a lot of the oxidation. You can see these carbons already have lost a lot of their electrons, and now they're bound to oxygen, which is going to kind of like take those electrons from them. By actually passing the electrons that it has, though, off into various carriers, then you're able to release little bursts of energy. And I'm talking about little bursts of energy because, well, if we go and we look at compare reduction potentials, we can compare these, and we'll talk more about how we can compare these using this E prime naught value. But bottom line is that if something has a high positive value for its reduction potential, it really wants those electrons. There's a high reduction potential. So we can see like oxygen really wants those electrons, whereas the hydrogen, not so much. Got a low reduction potential. And so if you were to pass electrons from the hydrogen to the oxygen, boom, you get energy. You often don't want to just have a big boom of energy. Often what you want to have is kind of like more piecewise pass-offs. And so we have electron carriers along the way, things like our NAD, things like our FAD and our cytochromes that can actually go and carry the electrons from one place to another, releasing the energy in little kind of like spurts. This is going to allow you to use the little energy bursts to pump out protons in your electron transport chain and create a gradient that you can then use like a dam in order to power the production of ATP. We'll talk much more about this process of electron transport chain and oxidative phosphorylation, but it's happening in your mitochondria. And your mitochondria are really cool. They're these little compartments inside your cell that come from a really ancient precursor of you swallowing a really old ancient prokaryote, it's like a bacteria. And then the ancient pre-eukaryotic cell is now a eukaryotic cell, and it decides to kind of hijack the bacteria and convert it for its own uses. Gets rid of a lot of the stuff, but keeps the energy making processes. And so you have, because it came from like swallowing that bacteria, you have this compartment, this inner compartment. This is the mitochondrial matrix. You have this inner membrane space, and then you have this outer mitochondrial membrane. So inner mitochondrial membrane, outer mitochondrial membrane, inner membrane space, and the mitochondrial matrix. But the electron transport chain is in this inner mitochondrial membrane, and it's going to be pumping protons out into this intermembrane space. What's going to happen is each of the little pass-offs from something that wants it less to something that wants it more, to something that wants it a little 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 more, to oxygen. Oh yeah, oxygen really, really wants those electrons. So what's going to happen is that each of those little pass-offs is going to have enough, give you enough energy to pump out protons. So you're pumping protons from the matrix into the inner membrane space that's between the inner mitochondrial membrane, where all these complexes are embedded, and the outer mitochondrial membrane, which is separating the mitochondria from the cytoplasm. Pumping out the protons using the energy from these pass-offs is building up the concentration of protons in this inner membrane space. This is going to allow us to have an electrochemical gradient. So it's a chemical gradient because we have more protons in here than in here. And so we have more of this chemical species in this compartment than in this compartment. And it's an electrical gradient because these protons are positively charged. So we're having a separation of charge as well as a separation of this chemical species. This is going to give you a strong electrochemical gradient. Gradients give you energy to move. And so we're using the energy from the electron pass-offs to kind of convert that energy into the potential energy of these protons flowing back through into the mitochondrial matrix. We're only going to give them one path through. That path through is going to be through this ATP synthase protein. Flowing through this ATP synthase, sometimes called your complex 5, well, this is going to be like a dam, like a hydrolytic dam. You have this flow of the protons through this dam that's going to be powering the turning of this ATP synthase, which is going to then allow for the production of ATP, which is a very energetically costly situation, but the energy from this gradient is able to pay the cost of doing that. We're going to talk much more about this chemiosmosis, so the movement of this chemical and the production of ATP through this like oxidative phosphorylation. We're using the power from these oxidations to phosphorylate ADP to give you ATP. We'll talk much more about this in next lesson, but today we're going to focus on the electron pass-offs. So these little pass-offs from something wants it less to something wants it more to more, pumping out protons along the way. And then we'll talk about using those protons to actually make the ATP. These electrons, remember, can be coming from all sorts of different fuel sources, which is why it makes sense to start from the bottom up, in my opinion. Speaking of which, we need to actually get those electrons here. And so that's another reason why it's important to have these carriers like our NAD and our FAD. So you want to get those electrons typically to the electron transport chain, but you're generating the electrons in various places. So for example, glycolysis, our breakdown of glucose, that's actually going to be happening in your cytoplasm. And so we need to basically get the electrons from it into the electron transport chain, and we can use carrier molecules in order to do so. Even if we're in the mitochondria, so say our citric acid cycle, which is actually where we're going to be doing the bulk of the electron, and spoiler alert, you can get there from a lot of different places, not just from glycolysis, but you're generating a lot of NADH, you're generating FADH2, and all of those are going to be able to feed into the electron transport chain because those NAD, that FAD is going to be able to carry those electrons for you. There are also going to be other carriers we're going to see, such as Q and various cytochromes. So we'll talk about these and how they can kind of allow for those passwise handoffs of the electrons. Because the amount of energy you get if you went directly from hydrogen to oxygen is the same as the amount of energy you get if you did it piecewise in these little pass-offs. And so, just like if you dropped a ball from the top of the stairs versus if you kind of rolled it down the stairs, you can have little bursts of energy versus one big burst of energy. Those little bursts of energy are going to be helpful for for the actual pumping of protons that you need to power the gradient. Because just generating energy isn't going to be useful for making ATP. You need to controllably release that energy, use it to do something useful. And so in this case, we're going to use it to pump out protons to make ATP. One thing, too, I want to draw your attention to is this complex 2. Note that complex 2 is kind of optional. You can take complex 2 out and still have this pathway running. Complex 2 is another way to Q. So Q is one of those carriers we're going to talk about. Complex 1 and complex 2 both pass to Q. And actually, complex 1 is optional too. You just need a way to get those electrons to Q. So complex 1 and complex 2 are two ways in which you can get electrons to Q, but we'll also see that there are other ways, such as from your lipid catabolism. The important part is getting things to Q, and then Q will take over from there. Complex 2 is actually your succinate dehydrogenase of your citric acid cycle. So when we talk about our citric acid cycle, this is the FADH2. This is your complex 2 right here. And so just a heads up, keep that in mind when you're thinking about the electron transport chain, is that the complex 2 is kind of optional, and that complex 2 is this succinate dehydrogenase from your citric acid cycle. Your citric acid cycle is taking place in your mitochondria, and your citric acid cycle is going to be one of the main locations where you are generating the bulk of your NADH, your FADH2. You are, however, going to be generating NADH from other pathways, such as glycolysis. But most of it's going to be coming from the citric acid cycle, and the citric acid cycle can be gotten to from a variety of different fuel sources, which is one of the main reasons why I think it's useful to kind of start from the bottom up. Let's start by thinking about the electron transport chain, and importantly, thinking about how the passing of electrons is used to generate this process, and how fuels, such as our glucose, such as our lipids, such as our amino acids, can actually be providing electrons, can be passing those electrons off through common carriers, and how those common carriers can then be used to power the production of ATP. Couple quick notes about mitochondria. We're going to be pumping protons out of the matrix into this intermembrane space. They'll then flow back into the matrix and generate the ATP on the matrix side, which can then get like swapped out so that it can flow out. This outer membrane is going to be much more permeable to things than this inner membrane. So this is going to make it so that we're going to be able to have things move more freely into this inner membrane space. But if you want things to go inside, well, now you're going to have special transporters, and this is going to come into play throughout. A quick note about mitochondria. So I've drawn them as kind of that simple cartoon, but mitochondria are far from simple. And they're not always a bean shape. So that was how people first saw it under a microscope. It looked like a bean, but mitochondria can look very different in different types of cells. Cells can also have different numbers of mitochondria. So our muscle cells, say, will have tons and tons and tons of mitochondria, whereas other cells that don't need as much energy won't have as many. Mitochondria can also do things like fuse together. And within a single mitochondria, you can have different amounts of kind of the folding of that inner membrane to give you these cristae. The more folding you have, kind of the more surface area you have to have those electron transport chain components embedded in them, so you can make more energy. In order to make all of those proteins that make up that respiratory chain, there you're going to have to make some of the proteins actually in the mitochondria themselves, and some of them are going to get brought in. So it's all this complicated stuff, but your mitochondria actually have their own genome, and it has their own ribosomes, and they actually make some of the proteins of the electron transport chain. In my postdoctoral work, so basically after I got my PhD, I went to another lab to do some more research and pick up some more skills and get some more experience. I worked on studying the effect of antibacterial antibiotics on mitochondrial ribosomes. So sometimes there can be side effects from taking antibiotics that target bacterial ribosomes because the mitochondrial ribosomes are structurally more similar to them than like our normal cytoplasmic ribosomes. So cool stuff on that mechanism wise, not side effect wise. But if you want to know more about it, I can tell you way more about it. Back to the story, however, of our electron transport chain, let's look a little closer at some of those carriers. So remember, a couple of the main carriers that we use in biochemistry are going to be our NAD and our FAD. So we have NAD plus gets reduced to NADH by taking that hydride, and we have FAD gets reduced to FADH2. There's also NAD plus, which gets reduced to NADPH. But that's going to be typically used more for our anabolism purposes. We're going to be using it as NADPH and oxidizing it to get us NAD plus in order to reduce other things. But of course, you have to be able to go back and forth with all of these, or else you're not going to be able to use them again. Let's think about some of the similarities and differences between NAD and FAD. I know that I'm going to be kind of lazy about saying like NAD plus or NAD. FAD, this is actually going to be tightly held in proteins. We call those flavoproteins. Whereas our NAD, this is going to be soluble so it can go and travel around. The FAD is typically going to have a higher but variable reduction potential than our NAD. If it has a higher reduction potential, if we were to pass something to it, we would get more energy. And so we can use it to drive oxidation reactions that require more energy. Things like, say, making a double bond from 2 carbon single bonds. Whereas with the energy needed to, say, go from an alcohol to an aldehyde, for that we could use NAD. But for going the harder reactions, we needed to use FAD. But if we use FAD, well, now we're at a higher reduction potential. And so we'll get less gain when we pass it off to the next thing in the line. The FAD reduction potential, though, is going to be variable because it's tightly held in the protein, and that's going to, the different proteins will have different environments that are going to change the actual reduction potential, but it's typically going to be higher than your NAD. But those are far from the only electron carriers. There are 5 main types of electron carrying molecules. So we talked about our NAD or NADP. We also have our flavoproteins. And so we talked about FAD, but there's also this similar molecule, FMN, flavin mononucleotide, as opposed to flavin adenine dinucleotide, also a flavoprotein, so also very tightly held. And that is a molecule that is going to come into play in complex 1, as we'll see. Ubiquinone. So this is also known as coenzyme Q or Q. This can move through the membrane. And then we have cytochromes, which are proteins with iron-containing heme groups, as well as iron-sulfur proteins. And so we'll talk a little bit more about these different ones. So remember when we talked about NAD and we talked about FAD, those were transporting pairs of electrons. Those were doing hydride transfers, remember? So we had that pair of electrons that was moving. When we talk about some of these metal carriers, when we talk about iron, when we talk about copper, these are going to take 1 electron at a time. And so we need a molecule that can take 2 electrons and give 1 electron. And it turns out that coenzyme Q is really great for this. It's got this little group where the action is going to be taking place, but then it also has this long hydrophobic tail. This long hydrophobic tail is going to allow it to hang out and move around the membrane. So that's going to be important in the CoQ-Q's role of transporting electrons from one place to the other, that it's able to move through the membrane. You might have heard of like Q10 supplements. Basically, that 10 refers to there being 10 carbons in this chain, this R chain. But we're not going to focus on that chain right now. We're going to focus on where the actual oxidation and reduction are taking place. In ubiquinone, so our Q, this is going to be the fully oxidized form. Then if it takes an electron and a proton, well, now we get this stabilized free radical. So remember, a free radical is when you have a lone electron. So not a lone pair, but an actual lone electron, unpaired, This is going to be our semiquinone radical, or our UB semiquinone. And so you'll see it drawn like this. But it accepts another electron and proton. Well, now you're going to get the fully reduced form, which we call ubiquinol, or QH2. So this is the fully reduced form. It required 2 electrons and 2 protons. And therefore, it's going to be able to give and take 2 electrons and 2 protons. And we can use it to both transfer protons as well as to transport electrons. Ubiquinone, the name kind of suggests it's ubiquitous. It's going to act as a sort of universal carrier because of its role of being able to accept 1 or 2 electrons, and it's going to be able to take electrons from both our NADH as well as our FADH2 or our FMN. What about iron? So iron, we can have that like +3 state, that +2 state. It can give and take electrons, but it needs an escort. We need to keep it safe because we don't want things giving and taking electrons willy-nilly, or else we'll get unwanted radicals. So radical in the case of our ubiquinone was helpful because it's just this transitory thing. It can actually, if it escapes, it can cause oxidative stress, oxidative damage. When you have free radicals, those are often very reactive. And therefore, when we use metal ions that do give and take electrons a lot, things like iron, things like copper, we're going to use them in a tightly controlled manner where we have them held very tightly in things like proteins. And even within proteins, we'll have them held tightly, such as in prosthetic groups. So these tightly held molecules like heme groups. Also good for holding onto iron are like cysteines. So the sulfur, so this would be like an iron sulfur cluster, as well as you can have them coordinated in these things called like Reski proteins, where you have them coordinated by histidines. So various ways we can hold on to those metals and take advantage of their ability to give and take electrons. So we'll see these different molecules be used, things like our cytochromes, so our proteins that hold on to one of those heme groups and that holds on to the iron. We'll see our ubiquinone. We'll see oxygen ultimately, and we will see this NAD and the FAD. All of those are going to come into play in our electron transport chain. Now we need to look in a little more detail about how this chain is going to work. I think it's best to start with kind of getting an overview of what it all actually looks like. So there's this great animation from the Harvard X. And so why don't you go watch that, and then we'll discuss in detail. All right, so we've got these 4 complexes, and really complex 2, we can take that out and it wouldn't matter. It would matter in the terms of our final yield. But complex 2 is really just an alternative way to give electrons to Q. So remember Q, that's our ubiquinone. We can give electrons to it from things like NADH, which we do in this complex 1, or FAD, which we get indirectly from succinate, which is complex 2. There are also other ways we can get things to Q, and we'll discuss that later. But the role of complex 1 is to get those electrons to Q. The role of complex 3 is then to take those electrons from Q and pass them on to cytochrome C, this little protein with one of those heme molecules that's holding onto the iron that can hold onto the electrons. And then the cytochrome C is going to go, and it's going to go to complex 4, which is going to pass those electrons onto oxygen. And then this is all in these complex 1, complex 3, and complex 4. Here you're going to be pumping out protons, and the pumping out of protons, then we'll talk about how we can create ATP. But first, let's look in detail about how each of these complexes work. So in the case of complex 1, this is also called NADH ubiquinone oxidoreductase, or NADH dehydrogenase. We're removing electrons from NADH, so we're oxidizing it, and we're reducing ubiquinone. So we're passing those electrons onto ubiquinone. But we're not doing that pass off directly. Instead, we're going to do it through a series of electron transporters. This starts with FMN. So remember that was one of those, this is then like a flavoprotein. So FMN was our flavin mononucleotide. It was like our FAD, but slightly different. And it's going to be held in this complex 1 protein. It's then the electrons are going to be transferred from that through a series of our iron sulfur centers, ultimately to our coenzyme Q. Each of these pass-offs is going to be energetically favorable because we'll be going from something with a lower standard reduction potential to something with a higher standard reduction potential. We're going to couple the energy from those favorable transfers to the pumping of protons. So for each NADH that goes in, you're going to get 4 protons pumped in complex 1. And you're going to be reducing 1 Q molecule, giving it 2 electrons. So you're fully reducing it to its ubiquinol form. So we went from ubiquinone to ubiquinol. We now have this QH2, this ubiquinol, that we can use to pass energy on further. But first, before we go back to our story about the ubiquinol, we need to talk about another way we can form ubiquinol. And that's going to be our complex 2. So remember that complex 2 is your succinate dehydrogenase of your citric acid cycle. Sometimes called your tricarboxylic acid cycle, your TCA, your Krebs cycle, whatever you want to call it. Complex 2 is this succinate dehydrogenase. It takes succinate and passes electrons from succinate onto FAD to give you FADH2 and generate fumarate, which goes and keeps going on the cycle. Most of the citric acid cycle is happening inside of the matrix, but this complex 2 is embedded in that inner mitochondrial membrane. This, therefore, directly passes those electrons into the electron transport chain. So that complex 2 is going to then take these from FADH2 and pass them on to Q. That'll regenerate the FAD, which can do this again. And you now have your electrons in the chain to go make some ADP. And complex 2 being in the membrane is really important because FAD, remember, that needs to be tightly held in a protein. And so we want that protein to be in the membrane where the electron transport chain is. Because the FAD can't just go float around to Q like you could have in our NADH. Instead, we need to have the protein near where Q can actually access it. So it's going to pass the electrons from succinate to FAD, then through some iron sulfur centers to ubiquinone to give us ubiquinol. What's a little confusing is, yes, we're going to be transferring these electrons, but we're not actually going to be pumping any protons. So you might wonder, like, well, what's the point? Well, the point is to get the molecules to ubiquinone. So our ubiquitous carrier, that thing that's going to be able to transfer those electrons further down the line, we need it to be, the electrons to be there. So we're going to waste the energy from this in order to do that. Waste in the terms of we're not pumping out protons, but we're still doing something very useful. Why aren't we pumping out protons? Remember that our FAD had this higher reduction potential, which means that it's going to have less of a difference of reduction potential between it and Q than there would be between NAD and Q. And therefore, we're not going to get as much energy from this pass-off, but we still are going to get that ubiquinol. Now that we have that ubiquinol, let's go and let's take it further. Let's use it to transfer those electrons to cytochrome C. So cytochrome C, remember, that was going to be kind of like our relay protein. It's going to be this small little protein that can move around this inner membrane space. And it's going to be able to take those electrons from ubiquinol and transfer them to the 4th complex, which is where we're going to be transferring them to the oxygen. Complex 3 is called cytochrome BC1 complex, or ubiquinone cytochrome C oxidoreductase. So these long names. Complex 3, a lot simpler to say. And it's going to pass electrons one at a time from our ubiquinol to cytochrome C. So remember, ubiquinol is fully reduced. It's got those 2 electrons it can transfer. Cytochrome C, it's going to only transport 1 electron. So cytochrome C, it's got that team group that has the iron, and the iron is only going to be giving and taking one electron at a time. So we're going to need to just give cytochrome C one electron at a time. But we have 2 electrons in our Q, in our ubiquinol. And so we don't want to waste that second electron. Instead, we're going to go through some complex gymnastics in order to take ubiquinol and pass one of the electrons from it to the cytochrome C and kind of hold on to that other electron until we go to a second cycle. So there's this thing called the Q cycle. I don't need you to worry about the details. Bottom line is we're going to be passing those 2 electrons from QH2 onto 2 cytochrome Cs and pumping out 4 protons. But it's not going to be as simple as just doing it straight like that. We're going to do it in a couple of cycles. Now you've got those cytochrome Cs that have those electrons. Complex 4, and we're going to take those electrons from cytochrome C and pass them on to oxygen. Oxygen wants them really, really badly, remember? And so this is going to be highly energetically favorable. We're going to use it to drive the pumping of protons again, because we get that big boost, we get that protons pumped. And in the process, we're going to be making water. The numbers get a little weird here because when we think about reducing oxygen, well, oxygen, molecular oxygen hangs out as O2. And so we're actually going to take O2 and we're going to add 4 electrons to it. So we're going to need 4 cytochrome C molecules in order to do this. And we're going to create 2 H2Os. In the process, we're pumping out 4 protons. But if we were to think about it from the perspective of that initial NADH that was going into the line or something like that, well, then it would be the equivalent of 2 protons. So 2 protons per oxygen, but because it's molecular oxygen, it's O2, then it's going to be 4 protons overall. How this transfer is happening is it's going to be happening through several carriers. These include cytochromes A and A3, as well as copper ions. So you've got this kind of like iron and copper working together. So complicated transfers, but ultimately you're basically the same story over and over. We're passing electrons from something with a lower reduction potential to something with a higher reduction potential, generating energy, using that energy in this case to pump out protons and boost that proton gradient. Putting it all together, we can think of these 4 complexes. We have complex 1. We're going to take electrons from NADH, pass them off to Q. This is going to take our ubiquinone and give us ubiquinol in our Q pool. We can also give electrons to Q through succinate dehydrogenase or complex 2, which is the same succinate dehydrogenase that we had in our citric acid cycle. This is another way we can give electrons to Q. And it turns out that there are actually a lot of different ways to pass electrons to Q, including from things like fatty acid oxidation. So in fatty acid oxidation, we're going to need to do one of those reactions that requires the formation of a double bond and between 2 carbons. So we're going to use our FAD. And this is actually going to then contribute directly those electrons into this Q pool. So we talk about this kind of like general Q pool because it doesn't really matter where those electrons came from that are given to Q. Now you've got that reduced Q, you've got your ubiquinol. So wherever the source of the ubiquinol, now it can come and it can enter this complex 3. In the complex 3, those electrons from ubiquinol are going to be passed to cytochrome C. Cytochrome C, this is this little protein that can move in this inner membrane space. It's going to go and travel to our complex 4, which is where those electrons are then going to be passed off to oxygen, giving us water. In complexes 1, complex 3, and complex 4, where we get enough of an energy boost from those electron pass-offs in order to pump protons out. But with complex 2, here we don't. There's not as much of an energy difference in passing those electrons from the FAD to Q, then from NADH to Q, we don't have enough energy here in order to drive the pumping of protons, but we are able to contribute to this Q pool that we can then use to pump protons down the line. So if we think about electrons entering from NADH, well, for each NADH, we're going to pump out 4 here. We'll pump out 4 here, and we'll pump out 2 here. And so that would be the equivalent of 10 protons pumped out per NADH. And remember here, this is looking at it in terms of a single oxygen, so half an O2. So that's why you see this 2 protons as opposed to 4 protons pumped out, because we're talking about in terms of this kind of like half of the reaction happening. In the case of if we were to start with our FAD, if we were to start just by adding something to Q through one of these ways that doesn't directly go through complex 1, well, now we're only going to have these 6 protons pumped out. So when we come to accounting later on, we'll have to keep that in mind. We're getting more protons pumped out if we enter at complex 1, but we can also enter at complex 2 or any other route to Q. And although we kind of look at these as these isolated things, actually these complexes often hang out in these kind of bigger groups called respiratomes. So the complexes themselves are often very complex and they have lots of different proteins. And then they hang out together in these respiratomes where they're kind of close together in the membrane, which makes sense because, well, you don't want those electrons flowing all over the place. To review, we can break down all sorts of different fuel sources through oxidative reactions in which we pass the electrons from the fuel sources onto electron carriers, our NAD and our FAD, and we can transfer the electrons from those through a series of electron carriers in the electron transport chain. By passing electrons from something with a lower standard reduction potential to something with a higher standard reduction potential, we're releasing energy. That energy is used to power the pumping of protons. The protons are pumped into this inner membrane space. This creates an electrochemical gradient. The power of this electrochemical gradient is used to power the synthesis of ATP through a process that we'll talk about in next lesson. Through these series of carriers, we're able to generate this gradient. We have 3 places in which we pump protons, and then we also have carriers between them. NADH comes in at complex 1, or C1, and you pump out 4 protons. The electrons get passed from NADH through carriers onto Q, and then Q takes them onto complex 3, which pumps out another 4 protons, passes the electrons to cytochrome C, which then brings them to oxygen in complex 4, which pumps out another 2 protons. Note that we bypass complex 2, because complex 2, well, that's just another way to Q. Complex 2 takes electrons from FADH2, and this complex 2 is actually succinate dehydrogenase that we'll see come up in the tricarboxylic acid cycle, our TCA. So there's actually a lot of different ways to Q, and so we can actually take out complex 2, and this pathway would still work. We could take out complex 1, and it would still work. We just need a way to get those electrons to Q, which can get them to complex 3. If we come in at complex 2, however, we don't pump those initial 4 protons, and so we don't get as much ATP being made because we're not going to generate as much of a gradient. The gradient is going to be what we're going to get the power for this ATP synthase from, and that gradient is coming because you're passing the electrons from things with lower standard reduction potential to things with higher standard reduction potential.